Okay, so in this lecture video, we're going to take a look at how we can use Lineweaver Burke plots to get some information about the mechanism of an enzyme catalyzed reaction. Now, in terms of mechanisms, I'm not talking specifically about, there's a nucleophilic attack here and this proton moves over here. I'm not talking about that detail of a mechanism. I'm talking a little bit more just about how the enzymes and substrates are interacting with each other and in what order. And there are two mechanisms that we can distinguish from one another utilizing a Lineweaver Burke plot. One is an enzyme mechanism called sequential that involves, a sequential mechanism that involves formation of a ternary complex. So a ternary complex is just what it sounds like. It's an enzyme bound to three things. Okay, and there are going to be two types of ternary complexes that can form, and these reactions that we're talking about are going to have two substrates. So we're talking about catalyzing a reaction between two different reactants. So in all of our past examples so far, we've been taking one molecule and converting it into a new molecule. So in all of these mechanisms, what we're talking about are multi-substrate mechanisms. So for the formation of a ternary complex, which is a sequential mechanism, there are two kinds. The first is a random kind, and it looks a little bit like this. So you have an enzyme, and that enzyme can bind substrate 1 and substrate 2. Actually, maybe it would help if I just wrote the whole thing out like this. Enzyme plus substrate 1 plus substrate 2 eventually goes to enzyme plus product 1 and product 2. So in this sequential mechanism, we take our enzyme, and in the random model, the enzyme could either bind substrate 1 first, or it could bind substrate 2 first. It doesn't matter, it's random. Then the enzyme binds the second substrate, whichever one that is, and that's the formation of the ternary complex. So that's where we have the enzyme bound to two substrates. Then the chemistry proceeds, and you end up with the enzyme and the two products. So this is a sequential mechanism where you have the formation of the ternary complex, and it happens in a random order. The other type then of sequential mechanism is an ordered sequential mechanism, which still involves formation of the ternary complex, but in this case, it's the first substrate that always binds first, whatever that one might be. So enzyme plus substrate 1 forms the first complex, enzyme substrate 1. Then the second substrate comes in, and you form the ternary complex. And then the chemistry happens, and you end up with those molecules converted into products. So in both of these cases, we form the ternary complex, but it depends on whether or not the same substrate always binds first or if it doesn't matter. Okay, so that's the first type of mechanism, sequential. The other type of mechanism that we can use then to distinguish from is called a ping pong mechanism. And once we take a look at it, maybe you'll see why that is. And it does not have the formation of a ternary complex. So at no point do you have two substrates bound to the enzyme before chemistry happens. So the chemistry looks like this. You have the enzyme plus the first substrate. They come together to form a complex. Then something happens. The enzyme gets changed, and that first substrate also gets changed. So perhaps maybe a functional group got removed from the substrate and covalently attached to the enzyme. That would be a way that the enzyme could become changed. So that's why I put the prime here to indicate that it was a changed enzyme, so it's different. So at that point, that first product is released. And at that point, now we have the second substrate coming in. We form a second complex of this changed enzyme with the second substrate. And then the chemistry happens. The enzyme gets converted back to its original form, and you form your second product. So for example, we could be talking about transfer of a functional group from substrate 1 onto the enzyme, and then from the enzyme onto substrate 2. So that would be an example of a ping pong mechanism. So in this case, there's been no formation of a ternary complex. So we can use double reciprocal plots to identify if we have a sequential or a ping pong mechanism, and they end up looking something like this. So in a sequential mechanism, I guess I should say, the way you do these experiments is you're going to have a whole bunch of test tubes. So you're going to have your first concentration of substrate, number 2, let's say. We're going to talk about substrate number 2, and it's going to be A. And you're going to have a series of test tubes, and all of those test tubes are going to have a concentration of substrate 2 equal to A, and the concentration of substrate 1 will be increasing. Then you'll do a second concentration of substrate number 2, we'll call that B. You'll do another series of test tubes where again the substrate 1 concentration is increasing. Okay, and you do that for multiple concentrations of substrate number 2. So you're going to have a whole bunch of data points. Then what you do is you make a whole bunch of Lineweaver Burke plots, and they come out looking differently dependent upon if you have a sequential or a ping pong mechanism. So if you have a sequential mechanism, your graph ends up looking something like this. So 1 over the concentration of substrate, 1 over V0. So you get something like this. I didn't cross those very good. Pretend those all cross right there. So here's experiment 1, here's experiment 2, here's experiment 3. And as we move from experiment 1 to experiment 2 to experiment number 3, the concentration of substrate 2 is increasing. So that's an up arrow. So remember here it might be concentration equal to A from our previous slide. Here it might be equal to B. Here it would be equal to C, whatever those numbers are, but they're increasing concentrations of substrate number 2. Along the x-axis then are those 5 test tubes. So each one of these experiments had like the 5 test tubes from our previous slides. So this is the concentration of substrate number 1 across the x-axis. And so what we find here is we have an intercept. So if we see this, this is telling us that this enzyme mechanism involves formation of a ternary complex. So substrate 1 and substrate 2 are both coming together and binding the enzyme all at once. Now whether or not substrate 1 or substrate 2 binds first, that we can't tell. So we can't distinguish if this is a random or a sequential formation of the ternary complex, but we can tell it's a sequential mechanism. As opposed to a ping pong mechanism, if we did the exact same experiment and made the double reciprocal plot, so again on the x-axis 1 over the concentration of S1, it would look like this. So instead of getting experiments that cross each other, here's experiment 1, experiment 2, experiment 3. From experiment to experiment, in these cases the concentration of S2 is increasing. So in this case, we get parallel lines. So that tells us no ternary complex is formed. So it gives us at least a little bit of insight into how the enzyme is functioning for a multi-substrate enzyme catalyzed reaction.